Good morning, everybody. In this class, we shall be discussing about construction of recombinant DNA using NIF gene or nitrogen fixing gene. This is a sort of repetition of our first class. In the first class, we had discussed about the methods in recombinant DNA technology. So this is the same with reference to nitrogen fixing gene. So the topic is construction of recombinant DNA using NIF gene. As you all know, all living things require nitrogen. It is a key element of amino acids. What are amino acids? The building blocks of proteins. There is a huge reservoir of nitrogen in the atmosphere. Approximately 80% of the air consists of nitrogen gas. But plants cannot use atmospheric nitrogen directly to make protein. It should be first, the gaseous nitrogen in the atmosphere must be first converted or fixed into forms which the plants can make use of. Most plants obtain their nitrogen from natural sources in the soil, so it has to be fixed in the soil. Natural reserves of soil nitrogen are normally low, so nitrogen fertilizers must be added to increase plant growth. Now, nature also has got an alternative method of providing nitrogen to plants and enriching soil nitrogen resources. This is called as biological nitrogen fixation. Many members of the leguminous plants, such as beans, peas, soya beans, or alfalfa, leucaena, all have special ability to use biological nitrogen fixation or BNF to meet their nitrogen needs. So, what happens is the atmospheric nitrogen has to be fixed in the soil by some soil microbes, mostly these bacteria, nitrogen fixing bacteria, so that the plants can make use of this nitrogen. Either they fix the nitrogen in the soil, atmospheric nitrogen in the soil, and then the roots will absorb this atmospheric nitrogen from the soil and the plant gets its part of nitrogen. Or in these plants, in the leguminous plants, the bacteria fix the atmospheric nitrogen into the plant itself. So this is all called as biological nitrogen fixation. Thus the plant get their part of nitrogen. Now, what are NIF genes? These are nitrogen fixing genes. These genes are responsible for nitrogen fixation and are found in both free living nitrogen fixing bacteria and in symbiotic bacteria associated with various plants. Free living nitrogen fixing bacteria fix 30% of atmospheric nitrogen in the soil, and symbiotic bacteria fix 70% of nitrogen inside the plant itself. The NIF genes are genes encoding enzymes involved in fixation of atmospheric nitrogen into a form of nitrogen available to living organisms. In symbiotic bacterium, Rhizobium leguminosarum, the NIF gene is present near nod genes on the megaplasmid. There is a plasmid called megaplasmid. On that, there are some genes called nod genes. So, near those genes, the NIF genes are also present in the symbiotic bacterium Rhizobium. But in non-symbiotic cyanobacteria, the NIF genes are present on the main DNA, on the main cell DNA. Initially, NIF gene has been transferred in E. coli, Escherichia coli. The primary enzyme encoded by the nitrogen fixing genes, NIF genes, is the nitrogenase enzyme or the nitrogenase complex, which is in charge of converting atmospheric nitrogen to other nitrogen forms such as ammonia, which the organism can use for various purposes. In higher plants, chloroplast, which is a cell organelle, this might have been originated from prokaryotes, they say. Therefore, attempts are made to transfer NIF gene into chloroplast. So, the easy availability of the ATP and NADPH2 in the chloroplast also makes them ideal recipients for this gene transfer. Besides the nitrogenase enzyme, the NIF genes also encode a number of regulatory proteins involved in nitrogen fixation. The expression of the NIF genes is induced as a response to low concentrations of fixed nitrogen and oxygen concentrations. So, low oxygen concentration, low nitrogen concentration helps in the expression of NIF genes. That time the NIF genes become active. The low oxygen concentrations are actively maintained in the root environment of host plants. The first rhizobium genes for nitrogen fixation and for nodulation, NIF genes and NOD genes, were cloned in the early 1980s by Gary Raukan and Sharon R. Long in a person's lab called Frederick Ausbell's laboratory in California. Now this is a pictorial demonstration to show how the desirable genes such as cry gene, that you need not concentrate here, and NIF gene is cloned inside Agrobacterium tumefaciens. Agrobacterium tumefaciens is once again a soil bacterium. NIF gene is from Rhizobium, a bacterium. It is put into Agrobacterium tumefaciens and then transferred into another plant. So here, as you all can see, this is a bacterium called Agrobacterium tumefaciens. This is a bacterium Agrobacterium tumefaciens, and here you are taking out the plasmid. What is this? This is TI plasmid. This bacterium is a soil bacterium. Agrobacterium tumefaciens is a soil bacterium which produces diseases in plants called crown gall diseases. So this bacterium has a plasmid which is called as TI plasmid. This is the TI plasmid of the bacterium. This is a bacterium called Agrobacterium tumefaciens. So it has a plasmid. This is the main DNA. This is the plasmid DNA. And this plasmid is called as TI plasmid or tumor-inducing plasmid. This bacterium causes diseases in plants by inducing tumors by the help of this tumor-inducing plasmid. In this plasmid, there is a small length of DNA called as tDNA or transfer DNA. So this plasmid, this bacterium, Agrobacterium tumefaciens, is a natural genetic engineer. What we do in genetic engineering, this bacterium does naturally, incorporating pieces of genes into another organism. So then I'll tell you more about this now. Now this is another bacterium called Rhizobium. So there is another bacterium this is not the Rhizobium. There is another bacterium, Rhizobium, from which the NIF gene is removed. So here what we do, we'll go like this only, step by step. So this plasmid is removed, and then the TI plasmid is removed. It has a small fragment of DNA called tDNA or transfer DNA. The plasmid is removed from the bacterium and tDNA is cut by restriction enzyme. With the use of a restriction enzyme, this portion of DNA, tDNA, is cut. And then what happens? See here, this is the tDNA is removed here. And then the third step, the foreign DNA is here. The foreign DNA is inserted into the tDNA of the plasmid. So what we do? We have a foreign DNA. Here the foreign DNA is either NIF or the cry genes, NIF genes, the portion bearing NIF genes. So the foreign DNA is cut by the same enzyme and foreign DNA is introduced into the tDNA portion of the TI plasmid. See, this is the tDNA portion of the TI plasmid. So here you have inserted this foreign DNA. So this is the tDNA portion of the plasmid. Here our foreign DNA or the NIF gene has been introduced. And then, now this is a recombinant TI plasmid. This is a recombinant TI plasmid. Means this is the TI plasmid having a foreign NIF gene in the place of tDNA, inside the tDNA. And then what happens? This recombinant TI plasmid is inserted back into a bacterium. See, this is also Agrobacterium tumefaciens. The plasmid is reinserted into a bacterium. Then the next step is, the bacterium is used to insert the tDNA carrying the foreign gene into the chromosome of a plant cell. Then this bacterium is made to infect the plant cells which are grown in culture. So these are callus. So callus which are being grown in culture. This bacterial suspension is put into this petri dish. So now there is callus here and there are the Agrobacterium tumefaciens. The Agrobacterium tumefaciens get into the plant cells, attack, infect the plant cells. And then what happens? Here, see the inserted tDNA carrying the foreign gene that gets incorporated into the genome of the plant cells. So the bacterium is used to insert, this bacterium is used to insert the tDNA, carrying foreign gene. So here you have the tDNA carrying foreign gene. This is the tDNA push carrying foreign gene. That gene is made to insert into chromosome of plant cell. Here, inserted tDNA. This is the inserted tDNA carrying foreign gene. This is the foreign gene. So now here it gets incorporated. These plant cells are grown in culture. See, like this. Now all these plants have NIF genes in all their cells. So this is the technique. I'll tell about this again in the end. Once again, I will explain to you this picture. Now let us come to the steps in the construction of recombinant DNA using NIF gene. The steps involved in the construction of recombinant DNA using NIF gene. The first step is isolation of NIF gene. Second step is integration of NIF gene in the vector. First you are taking out the NIF gene, then you are putting this NIF gene inside the vector plasmid and then cloning the NIF gene. This plasmid is put into another bacterial cell. Multiple copies of this bacterial cell is made and that is called as clones. All these are clones. Cloning the NIF gene. NIF gene inside bacterial cell is being cloned. And then introduction into plant genome. Then these bacterial cells are made to infect the plants so that the cloned recombinant DNA gets into the plant. And there is integration of the NIF gene into the plant genome. So introduction into the plant genome. Then culture of the plant cells into trees by tissue culture methodologies. This is it. So now I will tell you in detail once again the steps in the construction of recombinant DNA using NIF gene. So the first step is isolation of NIF gene. See, this is a bacterium called Rhizobium. This is a bacterium called Rhizobium. Rhizobium leguminosarum is a biological nitrogen fixing agent inside the leguminous plants. It is a symbiotic organism which gets into association with roots of higher plants, and there it directly fixes the atmospheric nitrogen into the plants. By making nodules, it stays there in the roots of higher plants, leguminous plants. So this has the plasmid, and in that there is NIF genes. So first we have to isolate the NIF gene from the bacterium Rhizobium or any bacterium, nitrogen fixing bacterium. Isolation of NIF gene. NIF genes are present in the nitrogen fixing bacterium Rhizobium here. The bacterial cell is opened by the enzyme lysozyme. There is an enzyme which is used to break open the bacterial cell. That enzyme is lysozyme. By treating this Rhizobium with the lysozyme enzyme, the cell wall is burst open. Then the plasmid DNA is treated with specific restriction endonuclease. It is treated with a restriction endonuclease so that it produces DNA fragments with sticky ends, and these fragments carry NIF genes. Okay, these fragments carry NIF genes. It is actually a cluster of genes, NIF genes. So like this we are isolating the NIF gene. So this is the first step wherein we isolate the NIF gene. And then integration of NIF gene in the vector. Next what we do, there is another bacterium called Agrobacterium, and it has a plasmid called TI plasmid. As I told you, the TI plasmid is isolated and it is cleaved or cut open at a site in the tDNA segment. I told you TI plasmid of Agrobacterium tumefaciens has a small length of DNA called tDNA or transfer DNA, and at this site in the tDNA segment there is a cut made, cleaved. It is cleaved with the help of the same specific restriction endonuclease. Now the isolated NIF gene is spliced or joined into the opening with the help of enzyme ligase. See, it is spliced with the help of enzyme ligase. Thus, NIF gene is incorporated into the TI plasmid. This NIF gene integrated with TI plasmid is called recombinant DNA. It is called recombinant DNA. So here also there are sticky ends, here also there are sticky ends, and so there is integration of the NIF gene into the plasmid. So NIF gene is incorporated into the TI plasmid. This NIF gene integrated with the plasmid, TI plasmid, is called recombinant DNA. Now, next step is cloning the NIF gene. As I told you, cloning the NIF gene. Cloning is a process by which hundreds of copies of NIF genes are produced. See, hundreds of copies of NIF genes are produced. For this, the recombinant plasmid is introduced into agrobacterium. See, this is agrobacterium. So, once again, it is reinserted into agrobacterium. As these bacterial cells divide, the plasmid and the NIF genes are also replicated. See, many number of clones of the plasmid and NIF genes. Thus, thousands of copies of NIF genes are produced. The next step is introduction into the plant genome. Next, introduction of the NIF gene into the plant cell or plant genome. Now, the bacterium containing the recombinant plasmid, these are the bacteria containing recombinant plasmid, is introduced into plant cell. It is introduced into plant cell. In the cell, what happens? The tDNA, this is the tDNA. The tDNA containing the NIF gene of the plasmid, the tDNA containing the NIF gene of the plasmid gets separated. See, it has got separated here and is transferred into the plant cell genome. It is transferred into the plant cell genome, where it gets incorporated with the plant genome and replicates along with the plant genome. Thus, the plant genome gets the property of nitrogen fixation. Thus, the plant genome gets the property of nitrogen fixation. So, this is a picture of construction of recombinant DNA using NIF gene and transformation of plant cell. Means it gets into the plant cell. We say the plant cell is transformed, transformation of plant cell. So, the fifth step, last step is culture of plant cells into trees. The plant cells, such plant cells which have the nitrogen fixation gene in them, integrated in their genome, such cells, plant cell is cultured by tissue culture methods and it is converted into a tree. All the cells of this tree contain the NIF gene and they can fix free nitrogen. So, here is a cell and this is culture medium in a test tube, and this is a plantlet developing and this plantlet becomes big. Hybrid containing, now this is a hybrid containing NIF gene. So, this grows into a normal plant.